Good evening. I'm Richard Murray, Chair of the Division of Biology and Biological Engineering here at Caltech. For tonight's lecture, I have the distinct honor of welcoming our attendees, both those here with us in person as well as those online, and introducing our speaker. Let me start by thanking all of you for joining us in person and online for tonight's lecture, which is part of the Watson Lectures Centennial Season. The series was created 100 years ago for the purpose of introducing and exposing the public to science, technology, engineering, and mathematics. And through public talks by faculty across the Institute's research divisions, the tradition has continued, providing Caltech faculty with an opportunity to share their research with public and the local community. I'd like to offer special thanks tonight to friends of the Beckman who help support activities and events around the lecture series, which help broaden the reach and access of these events. With their support tonight, we are hosting students from Anawaka-Mekak International University Preparatory of North America. Our speaker this evening is Magdalena Zernicka-Goetz, the Bren Professor of Biology and Biological Engineering. Magda grew up in Poland and received her PhD from the University of Warsaw in 1993. She did a postdoc for a few years in the United Kingdom and then started her independent research group at Cambridge University. She started her position at Caltech in 2019, just before the start of the pandemic. Magda has received numerous accolades for her research, including being elected as a Fellow of the British Academy of Medical Science in 2013, and election as a foreign member of the Polish Academy of Science in 2017. More recently, she received the 2022 Nomis Foundation Distinguished Scientist and Scholar Award. Magda's work is an excellent example of the type of work in biology and biological engineering here at Caltech. Paraphrasing a bit, Richard Feynman said, "What I cannot build, I do not understand." And Magda takes exactly this approach in her work. To understand the principles that underlie the processes involved in development, the dance of life, as Magda likes to call it, Magda builds systems that implement these principles and then studies them to provide insights into the natural world. It's a great fit with the division's approach to combining science and engineering to advance our understanding of the biological world around us. So with that, I'd like to introduce Magda, and we'll start with a short introductory video.
I was born in Poland, and my first memorable images are of people in white lab coats doing experiments. My parents didn't have a house or an apartment, so we lived for many years of our life in the institute where my father set up. And my father was a neuroscientist, and he was very interested in our feelings and dreams, where they come from. And I think that I found it also very intriguing, but then I became much more fascinated by the question of origins of these feelings, of origins of everything. And essentially this led me to ask the question how it is that one single cell can create all of that complexity. I refer often to it as a dance, and I wish to understand this multicellular dance. For a very long time, we all thought, and that's what was in my school books, that all of the cells initially are identical for quite a while. The first time that we realized that the cells at these early stages of embryo development, even after the first division, might not be identical is by painting them and finding that one cell will generate majority of the cells that will form placenta, while its neighbor, sister, will generate majority of the cells that will create the future embryo. It starts with some molecular asymmetries that will bias the fate in one or the other direction. But where those biases, where those asymmetries come from is still an open question in the case of the human embryo development. The knowledge that we have gained by observing real human embryos is allowing us now to build models from stem cells of human embryo. There are models. They cannot survive on their own and build a human being, but they would allow us to study specific processes that are responsible for certain stages and phases of development, steps, if you wish, that has to be really fulfilled perfectly for the embryo to go through. What we do with NOMIS Award, which is unique, is to build a structure that will be a reminiscence of the placenta. So by building this human embryo-like placenta structure and putting our stem cell model on it would allow us to look at the interface between the embryo and the mother without studying real human embryos. This was a black box of development, so we open up this black box to study this moment of implantations. This particular project has enormously important clinical implications because a majority of human pregnancies fail even before women know they are pregnant. Many people are coming from different disciplines when they join my lab. They are not necessarily working on embryos or stem cells when they're joining. We come from different countries, we have different backgrounds, we have a different way of shaping our thoughts, different strengths and weaknesses, and I love that. And by the way, I want to stress that weakness in science is very important. I don't want anybody in my lab to be perfect. In fact, I like those imperfections, and I think that's how we break the symmetry. I wish to start by thanking Richard for amazing introduction. This is really a great honor for me to be introduced for the very first time by you. And all the committee responsible for this series of very special lectures for allowing me to give one of them. It's really a great honor. And thank you all for coming here. I know it's much easier to watch that lecture from home by Zoom. So I was worrying that nobody will come, and I'm very happy that you came. So I would like to tell you a little bit about the beginning of our life, and I am not sure how you feel about it, but to me, despite that I'm studying it, it's quite incredible to realize that all of us come from one single cell that after a few days of its life make this collection of cells that actually undertake this incredibly intricate dance, like these real dancers on the right, to be able to touch each other, talk to each other in a chemical way, to build every single complexity of our body and mind. So if you think about it, it's really a mystery. So when you look at all of these creatures, they are spectacular from the mouse to human being. They have this unique shape, form, unique memories. They have their own social networks. So again, how it is possible that every single creature on that slide started in its life in exactly the same way? From this one single cell, a very powerful cell that created a ball of cells, and from this ball of cells, within a very short period of time, relatively short period of time, baby have to be created. So this is really what we are trying to understand in our laboratory. We are trying to understand the beginning of this journey, the beginning of this dance, and specifically trying to establish how this cellular choreography is determined and how embryos succeed and where and why some of them do not, and the pregnancies are failing and are terminated earlier than they should. So we are interested in addressing these questions because we are interested in the biology behind it, behind that mystery. But there is also important clinical application of that work, and this is because when you look at this pregnancy iceberg, you will see that only around 30% of the pregnancies are actually successful. Successful meaning that they result in the live birth. Majority of them are failing even before we know we are pregnant. So 30% of those failures happen within the first 7 days of our life, and the next 7 days is equally fragile. So again, around 30% of the pregnancies fail at the time when embryo invades the body of the mother in the process called implantation. So these are all of those preclinical losses that I would like to explain, and really I would like to understand what it is that is so hard for our embryo to get right. So that's the question. And to be able, this is the question that I would like to address today. And to be able to talk about it, I thought that I will split this lecture into 2 parts. In part 1, I will talk about how it is possible that this fertilized egg, within just these few days, is able to establish these 3 major tissues that are unique. And actually, I'm thinking about these 3 tissues, red, green, and blue, as these 3 dancers that have to be put on a stage at the end of the first week of human embryo life. So those tissues, they have the names, so I wouldn't refer to them much, but I will tell you that this red tissue is the most magical one in some kind of way. They are called, these cells of the tissue called the epiblast. And it's magical because these cells are the mother cells of every single cell of the future baby. The green tissue and blue tissues are really the ones that will not contribute to the embryo, to the baby, but actually they are the directors. They are like mothers and fathers of the baby. They are the ones that tell the red tissue what to do. So green tissue, with time, also become this sac called yolk sac, in which the baby will grow within the body of the mother. And the blue tissue called trophectoderm will become placenta to combine the baby with the body of the mother to survive. So the first part, I will tell you how we create these tissues and when things go wrong and what can we do about it. The second part of my talk will be about this another fragile stage of our life that is initiated by the time when embryo implants within the body of the mother. And this moment of implantation, until recently, we were not able to study because this is the time when embryo is still tiny. We cannot observe it in vivo. We could not mimic it in vitro, and the embryo become embedded within the body of the mother, and it's very difficult to recover. In human embryo case, we will never recover those embryos, but in mouse that we often study to understand human development, we could, but it's actually very difficult. So I will tell you how a few years ago we established 2 ways of looking at this stage of development soon after implantation. So I will start first with this part 1, and these are the 3 questions that I would like to address. First, how are these first 3 types of cells generated? How do embryos know to generate them on time? So how, without a real clock, embryo can measure time? And then what embryo can do about its future if things go wrong? So when I started to be enchanted by these early stages of our life, the dogma in the field was that initially, for the very first few days, when the egg cleaves into smaller and smaller cells, all of those cells will be identical for a very long time. They will be equivalent to each other until this moment, when embryo has 8 cells and transit to the 16 cell stage, when some cells will become localized to the inside part of the embryo, and those inside cells will be the one that will make either those mother cells for the future body or the yolk sac, and outside cells will initiate the journey for differentiation to form placenta. So, however, I have to say that despite that this was in the textbooks, we really didn't know whether this is entirely true. And it was questionable because in many other model organisms, many more simple model organisms, and embryos actually position factors that determine fate of the cells very early, already at the 2-cell stage. So when I started my group in Cambridge, I thought that it would be incredibly fun if we can paint those cells in such a way that those cells will not know that they are painted, so in a way that we will not disturb their mentality, and ask the question whether this is really entirely at random, how those cells contribute to those 3 different tissues by the end of this pre-implantation period of life. So we developed many different ways of painting cells. This was really very cool project, and I'm not going to show you all of them, but one of them was by injecting different markers, either fluorescent red or fluorescent green marker, and shooting movies of those embryos. So shooting movies now, everybody shoots movies, and I'll show you many more beautiful movies. But this was one of the very first movies that we shoot of mouse embryo development. So this is quite primitive movie, nevertheless was very influential because it showed us that actually cells start to have inclinations to become different from each other much earlier than expected. So if you take this embryo that have only 4 cells, 1, 2, 3, 4, this is tiny little marker that is not really a full cell. Then when you follow development of those cells in the embryo, you will find out that very often, although not always, which was confusing for a long time, there will be one cell that will have a tendency to give rise to majority of those cells that will form the future body. While other cell, its neighbor, will have a tendency to differentiate earlier, to give rise to the progeny that will make the placenta. So this was extremely unexpected. It was really against this feeling that the knowledge that I had and many people in the field had. So this was like a first unwanted discovery that I have made when I started started my independent path in the research. So it was so unexpected that we couldn't believe ourselves, and we did many, many tests of whether this is really true or not before we published. And then we thought that we can test whether those early differences are truly important by building embryos ourselves from one single type of cells. So maybe just build an embryo only from yellow cells or only from green cells and ask whether those embryos can actually develop to term. And here is an example of such a study. And I didn't tell you, but we know which cell is which by monitoring the order and orientation of how cells divide to create this 4-cell stage embryo. So when we monitor orientation order of the divisions from the 2-cell to 4-cell embryo, we will have this power of prediction which cell is which. And we monitor this orientation order in relation to this little marker, the polar body that is attached to the embryo. So now, when we take 4-cell stage embryo, fluorescently labeled, because those mother cells were labeled, we can separate individual cells from each other. And here I would like to show you this surgery. Embryos are tiny, at less than 100 micron of diameter. So you operate them with the very big machines. But you can see this operation is very beautiful. It's like a microsurgery. So here, what Carolina in my lab is doing, she's separating these individual cells from each other. You see here stills from those movies. And then she will put the same type of cells together based on these predictions of how the embryo was created at the 4-cell stage. And these experiments were incredible because what they showed us, the outcome was very important. They show us that when we build embryo, we call those embryos chimeras. When we build chimera consisting of those cells that give rise to majority of those cells that go inside and are tended to be pluripotent and make the body, those embryos do survive. Nearly all of them, 85% of them. When we put together embryo from those cells that are biased to form primitive endoderm, they would survive, but less frequently. While when we put embryo together from those cells that have a tendency to differentiate earlier and form the placenta rather than a baby, those embryos never made it. So this showed that those differences in cell fate are actually important. They are also showing that those differences affect the level of potency the cells have. So some cells are more equal than the others, are more pluripotent, the others are not. So we've made this observation, and of course this was a long time ago, more than 10 years ago, and we wanted, as typical scientists, understand what's the mechanism behind that discovery. What's the mechanism of how those cells become different from each other? When you look at them when you look at the embryo, you will not see any differences between those individual cells. They really look identical. So here we stopped this project for many, many years because there was no technology at the time available to take it any further. We couldn't really address the mechanism, so we started to do other things that I will tell you next about. But I wish to go 10 years forward and tell you about the technique that was established that would allow you to check which genes are expressed, which genes become activated in every single cell of that embryo. So we use this technique to ask, are those cells all show the same activity of genes that they would like to bring to life? So we actually did this single cell, it's called single cell RNA sequencing. We did it throughout the whole pre-implantation development, and now we are doing it also for older embryos. But here I focus on these 4 cells that we separate from each other, and we identify a pattern of gene expression. And the result was just very, very reassuring because we were able to identify many genes differentially expressed on a very high level of differential expression. So all of these little dots in red above this green line are those genes that are highly significantly differentially present or activated in these individual cells in the 4-cell embryo. So I don't want to go any further into specific actors that we picked up at the time to ask what they do. One of them you see is highlighted is SOX21. SOX is a big family of genes. We knew that some were important, but we never knew that SOX21 can have this very early role in making cell either more pluripotent if you have it, or less pluripotent if you don't. So this type of studies and many more that I'm not showing today allow us to propose the pathway of how cells become different from each other at the 4-cell stage that related to a particular enzyme called CARM1 that methylates DNA, actually methylates proteins that are bound to DNA, and this makes some proteins going and binding to DNA for longer, and they activate particular sets of genes. So in a way, you might think this mystery is solved, but actually it's not true. It's a big question that we still haven't addressed, is how all of this starts. What's the origin of this asymmetry? Why it is that at the 4-cell stage, one cell becomes different, as I mark it here in blue, another in green, and the other 2 are similar to each other. So the origin of this cell fate decision is still unclear. It could be entirely stochastic, so it could be at random that these cells at the 4-cell stage become different from each other at that point. But it could be also related to asymmetry within the egg that we cannot yet recognize, either mouse or human. Or it could be related to asymmetry that is imposed on the egg by sperm entry. Because after all, when sperm hits the egg, when it enters only on one side. So it might well be that there is some kind of memory of that side which received the sperm. So it's still unknown questions, but what I wish to say, very excitingly, the work of our colleagues, not our work, and I'm citing one of those papers here, showed that it seems that the same will be true in human embryos. So when we look at our organs in our body, when you are able to track them back through genetic marking that they accumulate when they develop, they seem to be going all to one cell at the 2-cell stage, not both. But whether indeed there are these early differences between cell setup in human embryo as what I'm telling are in the mouse is still unknown. Okay, so this is one of the interesting facts about this early embryo development, is that there is this heterogeneity between the cells, this uniqueness of cells that is initiated at the 4-cell stage. Now, I wish to stress that at that time, the cells are not determined yet what to make. They are just getting this inclination, like biases, and I think that's often the case how we develop too. Very rarely we are born with deterministic features of be this or that person, but we might have some kind of predisposition to go one way or the other about our life. So the next amazing feature about this very early embryo is happening just slightly later. So what I was talking now about is the embryo that has 1, 2, 3, 4 cells. So now when embryo has 8 cells, it's make another profound thing. And this is the polarization of every single cell within the embryo. When I'm saying polarization, this means that cells have to develop 2 different ends and acquire this amazingly beautiful apical domain on the outside layer. So you see here, when this embryo develops, the cells flatten against each other, that process known as compaction, and then those apical domains become established. And actually, we know about this already in the 80s. Now we can just make movies of this apical cap formation and go more into the mechanism of how it is set up. But it was in the 80s by Martin Johnson group discovered that embryos are doing this. So why do I talk about it? It's not just because it happened just after 4-cell stage, but because this acquisition of this apical domain is very important for cell fate. So if this cell, one of those cells, when it's polarized, cleaves into 2 cells, one daughter often will inherit that apical domain, and another daughter will not. And the cell that will inherit this domain will initiate the process of differentiation. So this is the cell that will tend to give rise to placenta. And this cell that will not develop as this pluripotent inner cell mass to maintain this magic of pluripotency to be able to create our body. So for a long time, we and our colleagues in the field were thinking how it is that embryo know that it should form this apical domain when it has 8 cells. It turned out that embryo has no idea how to count its number of cells. So it can't know, it can stop the divisions, and yet embryos with particular number of hours after fertilization will still form this domain, even if there are only 2 cells. So recently, we found that establishment of this apical domain and its timing depends on accumulation of 2 proteins to certain threshold. And I would like to show you because it's another clever mechanism that embryos develop, despite the fact that they don't have their brains at that stage, for sure not, and they are very simple, they are just created, and yet they develop this very intricate mechanisms to really set up everything in the right time and in the right place. So what we show is that, and this is Mengzu, the PhD student in my lab who published recently this paper, she showed that when genome become activated, zygotic genome become activated, those 2 proteins start to accumulate. They are called AP2 gamma and TIT4, and they have to reach certain threshold for the clock to tick. And this is because they would upregulate expression of activators for establishment of this apical domain. So I think this is another challenge for the embryo, or one of the first challenges for the embryo that I'm mentioning today, to go through to not be aborted. So essentially what I'm saying here, that there is this early heterogeneity between cells to incline cells to make one thing or the other. But when embryo has 8 cells, this is the 3rd day of the human embryo life, it has to put this apical domain. If it doesn't, it cannot proceed in development. And I think this explains many pregnancies being lost if this deadline is not achieved on time in our embryos. So the final mystery that I would like to talk about in this first part of the lecture is the embryoplasticity. So despite that the cell fate decisions are initiated relatively early, despite that we have this very strict developmental clock that have to be fulfilled, embryo as a whole is extremely plastic, which is incredible and actually very different to many other model systems, embryos of many other simpler model systems than mammals, including humans. So this plasticity was discovered experimentally. So when you have the embryo that have 2 cells and remove one, you see that very often one is enough to make a mouse. Not always it happens, but often is enough. This is quite an unusual feature of mammalian embryo development. And actually, this experiment was performed before I was born by my PhD mentor in Poland, Andrzej Tarkowski. He did another very nice experiment. He actually was the first to make chimeras together with Beatrice Mintz in US. So they were in parallel working on these chimeric embryos that I show you, but not the one that we created by making embryos composed of one single type of cells, but rather by bringing two embryos together and mix cells. So here they mixed two 4-cell stage embryos together, and they showed that these cells are able to communicate with each other in this process of dancing and form one single mouse that will have two types of progenitor cells. So both of those experiments show that embryos are quite plastic. So that's the question. Is this plasticity also used by the embryo in natural life when it develops naturally? So for example, can this plasticity of the embryo be called upon when there are problems with embryos? For example, some cells are abnormal. And I would like to tell you about the model that I developed only because this had happened in my pregnancy. My second pregnancy with Simon led me to establish this model that I call Simon now. So when Simon was an embryo, I took genetic testing on the third month of my pregnancy that showed that many cells, actually 25% of all of the cells tested in the placenta had genetic abnormality. And this was exactly the same genetic abnormality. There was extra copy of one of the largest chromosomes, chromosome number 2. So we all know about Down syndrome. This is chromosome 21, which doesn't have so many genes, and that's why often embryos with Down syndrome, embryos with additional chromosome that will have consequences and will develop into Down syndrome baby can survive. These news were bad because it's chromosome number 2, many, many more genes. So it was a cause of thought, for a thought, because obviously we now know that placenta that had this genetic abnormality and Simon are coming from the same mother egg, from the same collection of these very first few cells that I show you on the first slide. And of course, this is not just a matter of my own pregnancies. We know that so-called embryonic chromosome instability is responsible for low human fecundity. I already mentioned to you that 70% of embryos are aborted at the very early stage. Majority of these abortions are at the very early stages of pregnancy. And it's also responsible for a low success rate of IVF. So how we can test that? So when I was pregnant with Simon, I was wondering how I can live through this news and do something positive about them. And the way I was able to emotionally stabilize myself is to set up the series of experiments that would allow me to test the hypothesis, what happens to embryos that are mosaic, that have these normal and abnormal cells. So obviously, we cannot experiment on human embryos. We will never anyway know which cells in a human embryo are normal and which are abnormal. So to be able to do that, you will see we would be creating in my lab mouse embryos with chromosomal mosaicism. But essentially, I wish to tell you the 3 hypotheses that we had before we started these experiments. So let's imagine this is the human embryo that accumulates those abnormal cells as it develops through these early stages of development, egg to blastocyst. And those abnormal cells are red, orange, and blue, and those green cells are the ones that are normal. So 3 hypotheses. First, that those abnormal cells become eliminated. This would be wishful thinking. I would absolutely laugh if this is the case. The second possibility is that those abnormal cells are eliminated from that part of the embryo that will make the baby. But they're accumulating, they are not noticed, or even pushed, those cells to this lineage of cells, this group of cells that will make trophectoderm and then placenta. So this outside lineage of cells. And of course, the worst would be if actually those abnormal cells are not eliminated and they can contribute to both embryo, proper, baby, and placenta. So as I've mentioned, we would never be able to make such an observation on human embryos, so we have to create mouse embryo model to study that and check which of these three possibilities is correct. So the way we did it is to use a drug that is called reversing that generates chromosome imbalances during cell division. So essentially, this sort of cheats the checkpoint that every single cell has to make sure that the chromosomes are separating correctly during the cell division. So we treated some embryos. Maybe I will show you the rest of that slide to make it easier. So we treated some embryos. This one shown here in red. We had them pre-marked, so we will know which cells were treated and which not, with the drug at the time of the transition from 4 cells to 8 cells. Then we look by now DNA sequencing to make sure that indeed we are causing this aneuploidies in those embryos, those abnormalities. And here you can see one of those many reversing treated embryos. So every single circle here is one cell, and those segments in that circles are individual chromosomes. So here you see that only few of those chromosomes are diploid, so that means have a copy of mother and mother DNA. Majority of those chromosomes have aneuploidies. They are missing one, or they have extra chromosomes. And here is the control embryo that have and all chromosomes apart from this single one, because it's a male embryo, so it has only one copy of X chromosome, which is sex chromosome. The second is Y, and that's why it comes up here with one single blue segment. So now we make an embryo. Again, we make chimeras. So we take 4 red cells, 4 blue cells, make those chimeras, and monitor their development when they establish the structure in which the 3 lineages of cells, the 3 main actors, 3 dancers, have to be established. And this was really incredible. So this is just an example of many, many results. This is an example of a movie, stills from the movie that we saw during this experiment. It shows that the cells that become the red cells that we are treated with the drug, and we know therefore they are likely to have these abnormalities, will be eliminated by programmed cell death. Essentially, they commit suicide, but only in that part of the embryo that will make embryonic lineage that will make the baby, not in that part of the embryo that will give rise to this extra embryonic lineage, placenta. And this perhaps explains why so many genetic abnormalities can be detected in genetic testing in placenta. So it's incredible that this mechanism evolved in this way, and sort of, I'm not saying all of those abnormal cells are eliminated, but a lot of them are. So we actually ask the question, how successful development of those mosaic embryos, mosaic because they have normal and abnormal cells side by side, how many of them can develop to term? And what's the limit of number of abnormal cells that will still be tolerated and prune off? And here is the result. So here are normal embryos, all born. Here are all abnormal cells because all of that embryos were treated with the drug, all dead. And here is an embryo that is chimera, has 4 abnormal cells and 4 normal cells. All of those embryos are born normal. So as many as 50% of abnormal cells at the 8-cell stage in 3-day-old embryo, an embryo can correct it. Even when 75% of the cells within the embryo are abnormal, many of those embryos will correct themselves. Not all though, but many. So this is incredible because and again, it's still an open question how those cells know they're abnormal, whether they are truly committing suicide, as I said before, or is it prompted by cells that are normal, their neighbors, to prune those cells. It's still an open question. So what's the mechanism here? We still don't know. But clearly, embryos have this self-correct mechanism and can cure themselves. And indeed, Simon was born as a normal baby and now is growing, is even bigger than here on that slide. So either those abnormalities were only present in the placenta, or if embryo that we call Simon had abnormalities in that part that created his body, they were eliminated. So this is all what I wanted to tell you about this very first few days of our life. This was this part one about how embryo breaks its symmetry and cells establish their fate. I've told you how embryos polarize when they have 8 cells, and how developmental clock has to tick, and how clever they are to know how to measure time despite of lack of a proper clock. And I've told you about this plasticity that leads, that is actually used in natural development, I believe, for sure in mouse, but I also have a reason to believe this is so in human embryos. So the next stage of development was much more enigmatic, and I have to say that we know so much about these first few days of our life because both mouse and human embryos can be maintained in the lab, in a dish, with ease. They are very easy and very beautiful to study. But what happens next was this big developmental black box. The reason being is that when embryo reach that stage of development, when these 3 dancers are on a stage in the context of this ball of cells, those embryos to develop further had to be returned into the body of the mother. And it's true not just for humans, but also for mouse. So we knew very, very little about how these 3 dancers interact when they start to interact for the first time with each other. So until that point, they were mainly created. So there were not much dancing happening. It was just process of creation and putting them on stage. But now they dance. And I really wanted to understand this dance, and particularly because during these early stages of this post-implantation dance, the decisions are quite profound. So for example, one of the decisions is where to put the head. You know, which part of the embryo should make the head? Which part should make a heart, and why only one, not 2 heads, etc. So I was really very interested in the stage of development, and I will show you 2 approaches that we have developed with my group to enter this black box of development. So first approach was to establish conditions that would allow us to culture embryo at this stage when it has these 3 dancers in vitro. And we first did it for the mouse, and you see here one of those embryos developing in the culture dish. There are stills from the movie. And here it's an example of the study that, in which we try to understand, actually we try to look at the origins of those cells that decide positioning of the most anterior part of the body, the head. So now the question is, can we do the same for the human embryo? Legally, we can culture human embryos until day 14, so the end of the 2nd week. But practically, it was only possible to culture them for this first week. So can we go and develop a system that would allow human embryos, those ones that parents decide not to implant to the body of the mother, but the surplus one that otherwise would be damaged, destroyed, to use them for research and follow development of our own embryos, to understand what happens at this stage of our life and why so many pregnancies are terminated at the time. So here is an example of the human embryo that develops in culture. So this first movie, you will see embryo will move a lot because this is before it attaches. Before, normally it would invade the body of the mother in the process of this implantation, and this is the time when embryo will attach to the maternal uterine tissue. So you see here it moves a lot until it attaches into the substrate that we are using on the dish, and it starts to grow. And here is the second movie in which you see how those cells for the very first time start to proliferate, properly proliferate, not just cleave. What I mean by that it's for the first time embryos start to grow, rather than just create smaller and smaller cells. So this is a very, very good method, and many labs now, including, of course, our lab,
uses this method that we first master using mouse embryos to understand these early stages of our life. And here I'm showing this group of white cells that it's hard to believe, but every single of us look like that group of white cells when we were developing. And you see how they progress and how more and more of them come about every single day. And finally, this is the decision. This group of cells in green is also those sort of, this is this group of cells that sit outside the embryo but will direct the positioning of the head in the future. So this is the first system, the first approach that we developed to culture mouse and human embryos in vitro. So for the very first time, we were able to look at the interactions between these tissues and start to understand the choreography of this dance. So it is a very, very good approach, but has a huge minus. Minus is that it's very difficult to study embryos at the time. Of course, if you think about human embryos, very few human embryos are donated for research that are normal to be able to understand what happens to different groups of cells within the embryo and how they interact with each other and how they make these amazing decisions that they have to make at the time. But even for mouse embryos that we study in the lab mainly, it's very hard to follow this dynamic. So to really understand how this self-organization of these three tissues work that is clearly independent of the mother, because we can achieve it outside the mother, we developed this approach of looking, of modeling development of the embryo with stem cells. So at the very final minutes of the lecture, I would like to tell you about some examples of the models that we have established over the last few years to understand how cells interact with each other when they build the body. So that's the question. How do embryos generate order, as you can see here, from this chaos of individual cells? And this is the example of stem cells in culture that I'm showing here. It's not an embryo. So I wish to introduce to you a new concept of the stem cells. So you know now that embryo have these three type of cells, red cells, green cells, and blue cells, at the time of implantation. But what I didn't tell you is that you can separate those cells from each other and establish those cells as a stem cells, so the cells that will amplify themselves indefinitely in culture. So the most famous ones are these embryonic stem cells shown in red here. They were actually established for the first time by Martin Evans and Kaufman. Martin Evans was my postdoc supervisor in Cambridge, so I'm very fond of those cells. And also by Gail Martin, who worked in Martin's lab at the time. Trophoblast stem cells and Zen cells are the stem cells for this extra embryonic tissues. So trophoblast stem cells are stem cells derived from trophoectoderm, and Zen cells are stem cells derived from the primitive endoderm, and these were derived in the lab of Janet Rossant in Canada. So what I would like to tell you right now is about just few examples of the models that we put together by combining these different type of stem cells together in right proportions, and how successful or not successful we are. So actually the first model that we built took only embryonic stem cells, because all of the stem cell types have different requirements to be happy. So initially, we actually couldn't find the requirements that will make all of them happy to want to talk to each other and initiate body formation. So first model was a pretty primitive model, nevertheless very informative, only contain embryonic stem cells. So since we don't have these other two types of stem cells that normally will be present in the embryo, we dip these red embryonic stem cells in a gel of the extracellular matrix that normally will be secreted by these other actors. So we don't have it, so we provide it artificially. And as you can see here, these individual cells, they divide, and they form this three-dimensional rosette-like structure, in the center of which lumen will open. This is the pre-amniotic cavity. So here you see the colleague in my lab compared to the, again, dancing, but in the water. So you see here how these cells first are sort of not polarized and become polarized to form this rosette structure. It's three-dimensional rosette structure, much more difficult than synchronized swimming, because it's only one dimension, two-dimensional structure here. But this is really three-dimensional. I'm just not showing you all of the other cells. And then finally, there's a signal to open the cavity. So that's a very important moment in life of the embryo, because if something goes wrong, then multiple lumen opens, and that's really bad news. So those embryos will be aborted. So this is one model that models only fragment of the embryo, only this part that actually would later on form the body. So actually, I've forgotten about it. So I would like to show you how it looks in real life. So this is this group of unpolarized cells making rosette and opening the lumen. And now this is still ongoing work. We can make movies of those cells and try to understand how they come together and influence each other to make those three-dimensional structures. So that's what we are still trying to understand, and many other features that relate to this developmental transition. But can we go further? So to go further, since we are developmental biologists, we wanted to do it as it is done in the real embryo, by providing those stem cells for those two extraembryonic tissues. So first, we put stem cells for placenta, trophoblast stem cells. So here they are in blue. So you see red cells and blue cells, they come together, and after 3 days, they create embryo that it looks pretty good, but not perfect. We call it ET embryo because it's ES cells and TS cells together, but we of course have this amazing hero from my childhood, ET, so it was very appropriate. Oh, I have to say, editors didn't really like us to call this ET model, so we had to add one more letter. We call them ETS embryos, but actually, in my heart, they are still ETs. So here you see that by bringing the second compartment, we can now have much more sophisticated model. We can see how cavity opens in one compartment, in other compartment, and then how those two cavities come together to make amniotic cavity. And also this model, amazingly, leads to the moment of development where the second symmetry breaking moment happens to initiate process of body formation. But it's not perfect. Only 2 from 3 cells normally present. So as a final model, we add this third missing cell. And here's this model. So we now put together ES cells, TS cells, and Zen cells, and we totally change the design. So we don't use gel of extracellular matrix anymore because we actually want Zen cells to make it, and we want this to be made in an informative way that ES cells and TS cells will obey to its function, to its message. But we use this agrowell dish to put all of the cells on the bottom of the dish so they can easily interact with each other, so they can meet at the same table straight away, or the same dance floor, I should say. Still, I always think that I should stay with the same analogy, so dancers. So they should all be on the same spot to be able to interact with each other. So you see, when you get the structures and you follow their development, they are incredible. Many of them, but not all, look like real embryo. This is real embryo, 3 compartments, epiblast, trophoectoderm-derived extraembryonic ectoderm, then primitive endoderm-derived visceral endoderm tissue. And here you have ES cells, TS cells, and Zen cells forming these tissues that look indistinguishable. Sometimes they look even better, like this guy has something here and this doesn't. But most likely it is because it was attached to the mother, and this never was. This is entirely outside the body of the mother. So I would like to show you 2 examples of experiments at the very end of how we used so far those stem cell-derived embryos to really understand these early stages of development. So what did we learn so far? First, about the mechanism of how those cells recognize each other to build this amazing structure within the first few days. And second, how far they can go. So I would challenge them by saying, okay, what if they can just go to that point and then stop, and then they're not perfect, so they cannot develop any further. So again, we are going to shoot movies. So I already was showing you examples of the movies that we were shooting when we were looking at the very early stages of embryo development. So this was the very first movies of mouse embryo life. So now we use the same technology, but we look not at real embryos, we look at these embryo-like structures that we create from ES, TS, and Zen cells that are marked in different colors. So those movies are like a preludium for me for understanding the mechanism of cell behavior. You can see how many cells are engaging. You can see whether they exchange position or not. Are they immediately hooking up with each other, or they make the decision like chaotically? So this is really very informative to track those cells. We did it a little bit, but we haven't completed that. I wish to have hundreds of those movies. But Min Bao, who was doing those movies, was not that keen on making movies. So he did a lot of other things that are amazing, and I'll show you the next one. But movies is really something that we should still do to really understand better these different behaviors. But we had enough of them to realize that we have these two phases. First is sorting phase, when cells sort from each other, and then they stay in the positions and they make these compartments. So those kind of movies and many other studies in which we identify which proteins are present in one type of cells, not in the other, how we can change those proteins, led us to find that there are two critical principles of self-organization of stem cells into embryos at the time. The first relate to cell adhesion, and the second to cortical tension. So this paper was just published very recently, and Min won the cover for it, so I'm showing you here because it's very beautiful. So cell adhesion is really the factor that tells you how sticky the cells are to each other. And it turns out that these three cells, they have different code. They express different molecules that make them sticky in different ways. So that's how they recognize who is who. Cortical tension is really about softness or hardness of exterior of those cells. So again, it's very important in directing the cell's behavior. Now when we know, and I'm not going to go through all of those phases, but you can sort of see how those cells can touch and see, okay, you are like me, or you are a little bit like me, but not entirely like me, I want to have you outside or inside. So that's really what happens at the time. But this phase takes around 12 hours, up to 24 hours, so quick. Then when the cells are positioned, they stop moving, and they start to grow. So this is like they move from this fluid to more solid phase of development. So now we can also change those conditions and increase the efficiency of the self-organization process. So final part is how far those embryos can develop. So the first thing that we wanted to know is whether those embryos that are 6 days old from the time when we seed cells can undertake the process that we call gastrulation. It's like a milestone in our development. That's the time when we decide how we actually start to build the body. Everything that happened before gastrulation was preparation for building the body. So putting these dancers on a stage, making them interact, multiply, build the foundation for the body, break the symmetry to decide where they would put the head, where they would put the rest of the body, and now gastrulation is building. So could they undertake gastrulation? And the answer is yes. So you see this process by the cells leaving one layer, this epiblast layer, and moving between Zen cells and these ES cells that make this epiblast layer. So it's incredible, because if they can do this, and if they can do this properly, this means that they can really develop further. So this is actually the first model to our knowledge that is able to mimic all of those gastrulation movements of cells. So how far they can develop. So we actually, over the last 2 months, this summer, we published 2 papers that show 2 of those models that we created. They're slightly different, but I'm only going to show you some examples from one of those 2 models, in which we show they can develop as far as natural embryos in vitro culture. They develop to form the head, the heart, and the rest of the body. They initiate process of organogenesis. So you see here these natural embryos in culture. They don't look as this beautiful drawings of Charlotte, who is one of the first authors on this first paper. But you see that they are much black and white in real life, and these are those ETX embryos, now ES cells, TS cells, Zen cells. So we don't have them ETs anymore, they are ETXs. So actually, this is Charlotte's drawing. I wanted to show it today because I hope Charlotte is at this lecture. And we submitted it as a proposal for cover. We did not win, which I think is unfair. We didn't have it as a proper cover. But this is another model which we did set up by reprogramming of embryonic stem cells into these different fates. This one won. Well, they're both beautiful, right? Like, different journal, but thank you. So, but I'm going to tell you about this model, not that model, this part that Discover won. So first important question was, can they make the most anterior part of the brain, forebrain? This is very, very important because there are no other model systems created from stem cells that they can mimic development of the most anterior part of our brain. So this was incredible for us to see that in this ETX embryo model, we make the brain structure. As far as we can see, it's entirely normal. So here we look at the localization of the marker proteins that will mark different regions of the brain. And here you see the same proteins localized in natural embryos. So this is really opening the possibility, uniquely, to study development of the brain. And we are trying to now create brain organoids from those embryos. Secondly, they develop the heart. The heart-like structure, I should say, because maybe it's not, we see it very similar to natural heart, maybe it's not identical. So here you see again the number of proteins that we have been staining for to show this heart in a synthetic embryo, ETX embryo, and natural embryo. They form all of different chambers, and this heart is beating, as you can see here. I'm not sure whether I'm running this movie properly, but anyway, this is the heart structure that is functioning, it's beating. They form somites, which are really the block of tissues here on the back of our body that are very important for formation of all of those muscle here. So they form the somites in natural with the same numbers as real embryo at this stage of development. Uniquely, that's the last set of data today, they develop in that sac that normally embryos are, because with this yolk sac, because we provided Zen cells that form that sac. And this sac is actually the first site where the blood, the first blood that will form, will be on the blood island on that sac. So this is incredible, and we see here the marker that shows us that these blood cells are created in that model. So this is really the summary of what I've told you about in the second part of the lecture. But just to bring the first part of the lecture into that context, so I've told you a lot about how we create these three different cells, red, green, and blue, and how important it is to create them on time. So we have that clock to make sure that they are created on time. And it is so important because if embryo misses the window of implantation and does not have the right number of cells at the right time, this embryo will be aborted. So we learned that. We then learn by extending ability to culture embryo in vitro outside the body of the mother, mouse and human, how those three types of cells interact with each other. We learned their choreography, the signals they exchange to be able to form the proper foundation to initiate the process of gastrulation and make the body. So the knowledge of these stages of natural development help us to build the stem cell-derived embryos that are so faithful representation of natural embryos. So this is really something that I would say, if you asked me a few years ago, I would say it wouldn't ever be possible. So I am actually amazed that actual embryos can build themselves, because often we actually provide right environment, right knowledge, right factor, we make the cells happy, and they do that. We actually don't engineer them as such. This is the process of self-organization. And I think it's very important because we now understand so much more about the biology behind this process, that with time we would be able to correct the times when this does not go right and embryos become aborted. And also provide this information to IVF clinics to know which embryos they should select to transfer to the mother and how to take care of the embryo before transfer, that it has the highest chance of survival. I wish to stress also that by building the stem cell-derived embryos, we are also learning how organs are put together, how we create three-dimensional tissues. And therefore, the results of these experiments go beyond embryology, because we would be able to hopefully use this knowledge with time to repair organs in adult body. So I wish to finish with a very famous person and very famous quote, what I cannot create, I do not understand. So when I was recruited at Caltech, on one of the occasions, I came and there was this huge Richard Feynman portrait. I think there was, I guess, Matt had been an exhibition of his work. So Matt Thompson took this photograph of me with him. At that time, I didn't know I would ever be using it, but I thought it was very relevant because what he said, what I cannot create, I do not understand. Yes? Yeah, that's what he said. And now, when you think about it, we created those embryos based on our understanding of real developmental processes of real embryos, but we still do not understand them. I don't want to finish this lecture by telling you we now created them, therefore we understand everything. We understand some things that allow us to create them. But actually the magic of cells themselves, the magic of interactions between these three major dancers is still magic to us. So it will be many years, hopefully only many years, to really dissect it to the bottom of what exactly those cells are doing at each single time when they're creating those embryos. So I would say now I can create, so I can begin to understand. So at the end, I would like to thank you very, very much for listening to that journey, and thank every single person in my lab for being incredible, being my inspiration, and creating all of these different things. I didn't manage to talk about all of the individual projects that we carry on in the lab, but you can always contact us, and I'll be very happy to talk about current questions, many that I couldn't cover today, and future questions that we would like to address. I would like to thank all of the foundations that give us resources for this work, which I'm so grateful for. Particularly when we think about the stem cell-derived embryos, initially people said to me, there is no way we can do it. So I'm so pleased that some foundations trusted in that mission and were able to fund it. So I can talk about it to you today and share this knowledge. I wish to say that we would not have done it without amazing collaborations. Many of those collaborators are now at Caltech. We came here 3 years ago. And ex-lab members who contributed to this specific project, I would like to list them here too, and now they have their own labs in many different parts of the world. Thank you very much for listening. I would like to ask you to ask questions now. You can ask anything. I will try my best to answer. I wish to say that if I don't answer properly, just at the time of pandemic, this book came out that I wrote together with the science broadcaster, Roger Highfield, in England and more recently in US. So I advise you to look at that book because many nuances that I was talking today are much better explained, I'm sure, in that book.
Hello, yeah. I can start. I have questions from the online audience, and I think we have time for a couple questions overall. But the question was, do I understand this correctly, that 70% of pregnancies fail early, but huge numbers of genetic errors can be accommodated? This implies something else is causing the failures. Is that something you're exploring?
Yes, it's a very good question. So I wish to stress that it's predicted that 60% of the pregnancies are so-called preclinical losses. So even before we know we are pregnant. The 10% are miscarriages that are sort of clinical losses. So you are absolutely right. I mean, this question is absolutely right. So some of the reasons for failures are genetic abnormalities, but they are not the only reasons, because as we discovered, some of those genetic abnormalities can be eliminated, not by correcting them, but rather by eliminating the cells that represent them if there are other normal cells side by side. And if those cells can be awakening themselves to compensate for the loss, because of course they have to proliferate more to make up for the loss.
I'm Jerry. I'm Simon's classmate, actually. So it's kind of cool to see Simon up there. And yeah, I have a question about, like, last year we learned about designer babies. And like, I have a question if designer babies are the solution to solve all of, diseases, human embryo like problems. So do you think that's...
So first of all, I'm so pleased that you came. I have to say to Simon that you came. I didn't allow him to come. I was worrying that he would be intimidating me if he was here. So your question is very good. So, I often worry about the term designer babies, right? Because it implies that we really want to design them as we design our clothes or furniture. So I really worry about these terms because some people can twist it and use it in a wrong context. So I wouldn't call it designing baby, but rather choosing which of the embryos that are initiated naturally, for example in IVF clinic, have the highest chance to survive. That is what we would be able to do, and in fact we are already doing. We just have to enhance this ability further, because we still know that majority of the IVF procedures are unsuccessful. As you know, maybe you've heard that in some countries there is a tendency to have more boys being born than girls. And is this the question of preselection? This would be only possible in IVF clinic context, or is something else that is contributing to it? That's really, that I would not wish ever to design our future by preselecting boys versus girls embryos to be implanted. But eliminating genetic abnormalities, enhancing the ability of pruning cells that, as it happened, become abnormal, I would be for it.
Okay, thank you.
Thank you, Jerry. Yeah.
Hi, great talk, by the way.
Thank
so my
you.
question was that this behavior of the cells at the early stages, is there any effect of the environment on the behavior that you noticed?
Yes, so that's an excellent question too. There is effect of environment, and in fact, one of our current projects is to understand the impact of environment on metabolism of cells as they make those embryos. So we are interested in it because we know that what cells are surrounded by, they would be responding to, but also because in many cases, the environmental factors during the pregnancies would affect the outcome. So in that model, this is just incredibly powerful model to address that question. So which, for example, even simple things, which foods or drugs should be not taken by the mother who is pregnant, can be now tested directly on those embryo models. Just to give one example. But of course, there are also more, I would say, philosophical applications of understanding of that question, how we can manipulate environment to make cells most potent to do those things that we want them to do, and how we can stimulate environment in that direction. Yeah, I have a signal that we should stop here, and I wish to say thank you so much for coming, and it was a pleasure. Thank you.